today's session. Today we are going to discuss a very important topic, that is antiviral properties of the immune system. In this topic we will see that how our immune system and how the defense mechanism present in our body helps us to protect ourselves from various viral infections. Now, as all of you know that our immune system comprises of two parts, that is specific immune system and non-specific immune system. The specific immune system is also called as the cell-mediated immunity, and the non-specific immune system is also called as the humoral immunity. The cell-mediated system is governed by T cells, whereas the humoral immunity that is governed by the B cells. Among T cells, there are two different type of subsets, which are T helper cells and T cytotoxic cells. Whereas in case of B cells, there is the involvement of plasma cells, and these plasma cells later on help in the production of various antibodies. Now, as we know that viruses, they are basically a type of intracellular pathogens. Intracellular pathogens means whenever they attack a human host or human cell, most of the time they usually multiply within the cell and causes infection. For example, if this is the human cell, then this virus particle enters the human cell via a process called as the receptor mediated endocytosis. Our cell surface usually contains numerous of receptors which serve a very important biological process. Among this group of receptors, certain receptors acts as an anchor to which the viral nucleoprotein or the viral envelope spike protein, they can get attached to it and form a complex. And this complex, where the viral particle attaches with the receptor of the host cell, leads to the process of receptor mediated endocytosis. Once this receptor mediated endocytosis has occurred, now the virus, it gains the entry to the human cell. This virus gains the entry to the human cell. Now, in order to combat this type of infection, what happens is our immune system, especially the specific immune system and the non-specific immune system, combat the virus simultaneously by producing different type of mechanisms. These mechanisms usually involve first non-specific proteins like interferons. IFN or interferon. Now there are many different classes of interferon, and one of the class, that is class 1 interferon, has the ability to produce IFN alpha, beta, omega, and tau. Well, where omega and tau related interferons are produced in a very lower concentration as compared to that of the interferon alpha and beta. Now let us see that how this mechanism usually operates. Let's take an example over here. Like this is the cell, and in this cell we have various types of receptors, including receptor for interferon, or let's say here as interferon receptor. Now there is another type of receptor which has the ability to bind with the viral protein. Viral protein binding receptor. So this particular receptor binds the virus and when this virus basically binds to this receptor, it later on starts to get internalized by the mechanism of the receptor mediated endocytosis, which has been explained earlier. Now after it gains the entry, it starts to multiply and form its various copies, making the host cell infected. Now when this procedure is going on, the infected cell, it starts to release some sort of proteins, and this type of proteins basically acts as a messenger which starts to bind on the interferon receptors. And the proteins which are binding on the interferon receptors, they are obviously the interferons. Now what happens after the interferon has binded to the interferon receptor? Now whenever interferon binds with the receptor, IFN receptor, it basically eliminates the virus by three different mechanisms. The very first mechanism is activation of PKR pathway that leads to viral protein degradation. Second pathway is activation of RNase L that again leads to viral protein degradation. And then there is the third pathway, that is modification. In that particular modification, there is the change in the structure of the basic uh, nucleotide component, and due to the unrecognition of, or due to the failure in the recognition of the modified product, the virus mechanism is basically hampered. So let's see each one of these one by one. The very first one is the PKR pathway. So in the PKR pathway, what happens is PKR, or the protein kinase R, is normally present in the host cell. It is normally present in host cell, but in a very negligible amount. Okay, so whenever a virus has infected the system, okay, so yeah, whenever a virus has entered the system, what happens is there is the formation of dsRNA. Now this dsRNA acts as a catalyst on PKR and causes its autophosphorylation. So what happens is PKR in presence of dsRNA is autophosphorylated. After this thing has occurred, now the phosphorylated PKR targets the eukaryotic initiation factor 2, which is a part of the translatory mechanism. It transfers its phosphate group to this particular protein moiety, and once when it is phosphorylated, it leads to the inactivation of viral protein synthesis. So this is about the PKR pathway, which is basically initiated by the interferons. In the second pathway, what happens is the second pathway, here there is the formation of a complex. The complex is formed between interferon along with 2 prime 5 prime oligoadenylate synthase. So what happens in this case is this particular enzyme, oligoadenylate synthase, is basically a mixture of three components. The very first three components, the very first component basically includes 2 prime or 2,5 synthase, second 2,5 diesterase, and the third one endoribonuclease. that is RNase L. So the pathway works in a very simple manner. What happens is, for the activation of component 3, component number 1 and component number 2, both of them combine. Now when both these components, that is component 1 and component 2, combines, it becomes an active component, and this active component targets the RNase L, and when this RNase L is activated, it inhibits or inhibition of viral protein synthesis occurs. Okay, so this is the about the second pathway. In the third pathway, what happens is, there is the modification, and that modification is due to the enzyme called as the adenosine deaminase or adenosine deaminase, not diaminase, sorry for that. So interferon, it basically activates whenever the interferon is or interferon's concentration rises, it leads to the activation of this enzyme, that is the adenosine deaminase, and whenever this type of enzyme is activated, it targets the adenosine residue and converts it into ionosine, and whenever there is the modification of adenosine to ionosine, it leads to the deactivation of viral proteins. So in this particular way, in presence of interferons, the viral mechanism can be inhibited by the immune system. Now if we talk about the other defense mechanisms, so the other defense mechanism apart from interferon involves natural killer cell. These natural killer cells also combines with the interferons, and whenever the natural killer cell combines with the interferons, it can basically lead to a different type of activity. For example, like this is the host cell which has been infected by virus, and the cell surface or present on this infected cell comprises of various type of receptors or certain moieties. So those group of cells in which the viral protein has been synthesized and the complete viral structure has been formed, so their viral peptides are basically um, channelized outside, and whenever it is channelized outside, what happens is uh, binding of antibody takes place, and this binding of antibody or specifically we say the IgG or immunoglobulin G starts to bind in this area, and binding of the IgG basically signals, it basically stimulates or provides a signal to the natural killer cell. This is the natural killer cells, and these natural killer cells have various receptors which can target the Fc portion, because as you all know that an immunoglobulin comprises of two parts, that is the FAB and the Fc portion. So the receptors present on the NK cells have the affinity to bind on the Fc portion, and whenever they bind with the Fc portion of the immunoglobulin, they start to migrate, and that particular migration leads to its attachment on the infected cell. And finally what happens is this combination leads to the lysis of the infected cells. Okay, and this type of activity is called as ADCC, or the antibody dependent cell mediated cytotoxicity. If we talk about some other different type of mechanism through which the immune system can basically uh, protect us from various other uh, infection related to viruses, that will be covered in the next video. Thank you very much. If you like my presentation for this particular video lecture, don't forget to hit the like button and subscribe my channel for upcoming videos. Thank you very much.